Now, lately we were seeing the mechanism of action of enzyme. In that, we have seen what this lock and key mechanism is. The actually, when you talk about activity of enzyme, the enzymes may not behave in the similar way as we are claiming here. Whatever the models for enzyme action have been proposed by various different scientists in this field, all those models or theories, uh, they have been based on individual observations of that particular research. There are a lot more things to be explored regarding the activity of the enzyme. Currently, the most popular and most accepted models are either lock and key mechanism or induced fit mechanism. Lock and key mechanism is a rather rigid kind of mechanism where there should be 100% complementarity between the active side of the enzyme and the substrate molecule. As you see, uh, if there is even minor dissimilarity between the active side and the substrate molecule, there are chances that the reaction may not occur. To counteract that thing, another theorem has been proposed, which is referred to as this induced fit mechanism. As you could see here, this is your enzyme molecule coupling with the substrate molecule, and when it couples with the substrate molecule, there are a lot of changes in the entire enzyme molecule occurring here. Initially, active side is a bit different, a bit enlarged kind of active site. When the substrate attaches here, the enzyme compresses itself. It is similar that as if you are holding something in your hand. Say, if you refer to this model, say, just consider this is your enzyme molecule. My hand is your enzyme molecule. So, what does this induced mechanism says? That say this is the active side, the thumb, and this portion is the active side of the enzyme molecule, and suppose this is the substrate molecule. So if you see, as per the lock and key mechanism, the complementarity is not 100%. There is a bit of space remaining between the substrate and the enzyme molecule. So this induced fit model explains that short a bit of dissimilarity between the enzyme and the substrate molecule. What does it says? When such kind of things happen, when the substrate comes here, the enzyme brings about certain conformational changes. As you could see here, if I have to hold this fist in my hand, what I have to do? I have to compress my hand first. मुझे इस तरह से अपने फिंगर्स का अरेंजमेंट थोड़ा सा कंप्रेस करना होगा. Now I have grasped this fist. In similar way, same thing happens in case of your induced fit model also. That enzyme molecule brings about a bit of conformational changes in itself. As you could see here, यहाँ पर evident है. यहाँ पर अगर आप active side देखें enzyme की थोड़ी सी different है. और जब ये substrate के साथ couple होता है, so you see that active side is compressed as per the substrate molecule. Okay, so this gives the explanation to a bit of dissimilarity between the active side and the substrate molecules complementarity. Now, the next issue we have discussed before also, the activation energy. What is activation energy? The energy required to initiate a particular reaction to convert a substrate into a product molecule. Now, you could consider this thing, activation energy, as if that if you have to move something from one place to another place, what you do, you either push or pull that object. Okay, so when you are actually pulling or pushing that object, you are exhausting certain amount of energy to pull or push that object. Okay, if suppose there is one a table or a box, wooden box is placed on the floor, and if you want to move that box from one place to another place, what you have to do, you will just push that thing or you will just pull that thing. आप उसे धकेलेंगे या खींचेंगे, and during that process you exert a lot of energy for that purpose. Okay, now that energy required is referred to as the activation energy. Now, what our enzymes or catalysts are doing, that they are reducing the amount of that particular energy required to carry out the reaction. You could understand this thing, that say in case of that particular wooden box which you are moving, if suppose you place some kind of wheel or rollers below that box, what is going to happen? आपके पास एक बॉक्स है जिसके नीचे रोलर्स या कुछ व्हील वगैरह टाइप की चीज लगी हो, सो मूविंग दैट थिंग इज बिट इजियर इन कंपैरिजन अ बॉक्स विदाउट अ रोलर और अ व्हील. रोलर वाला बॉक्स मूव करना आपके लिए ज्यादा आसान है, कम एनर्जी आपको वहां पर रिक्वायर्ड हो. ओके, सो द सेम रोल which a roller or a wheel plays in case of a box, same role is played by an enzyme molecule. The enzyme molecule minimizes the efforts required to carry out a reaction. As a result, two things happen. The reaction becomes less energy consuming, and it also is accelerated. The rate of reaction also increases due to the involvement of enzyme. Now, one more feature of the enzyme is that although it improves the rate of reaction, it lowers down the activation energy, but it actually does not take part in the reaction. If you go to the previous slide, what you are seeing here, this is the free enzyme molecule. Now, this free enzyme molecule, this enzyme molecule takes part in the reaction. It couples with this substrate molecule and forms this particular ES complex. Okay, this ES complex is formed. There will be formation of product molecule. The product molecules are released here, and again the same enzyme is free to react with the other substrate molecule. Means the enzyme molecule actually is not being consumed by the reaction. It temporarily takes part in the reaction, carries out the reaction, and after that it is free to couple with the other substrate molecule. It is not really consumed during the reaction. It is used but not consumed during the reaction, and that is the best part of your enzyme molecule, that one enzyme molecule can couple or can react with n number of substrate molecules. Okay, this was the part related with the functionality of the enzyme molecule. The next important part here in enzymology is the nomenclature and classification of enzymes. When you are studying a substance, an object scientifically, you must know how to name that thing and how to systematically categorize that. For that purpose, there is one International Union of Biochemistry and Microbiology, which has recommended a method for nomenclature and the classification of enzymes, enzyme molecules. So based on that, every molecule will have two kinds of name. One is the trivial name, that is the common name or nickname in short. In simple terms, you can say the trivial name is the nickname of the enzyme molecule, and it will also have a systemic name, which is referred to as the official name of the enzyme, that will be universal. Trivial name can change from a researcher to researcher, a country to country, or a lab to lab, but systemic name will be universal. आप आप जब रिसर्च करते हो, you come across with one particular newer kind of enzyme, you may give whatever name you like to that enzyme, that is referred to as the trivial name. But after systemic studies, what kind of reaction the enzyme carries out, what substrates are involved, what kind of product is formed, based on that there will be a systemic classification of enzyme, and after that systemic classification, that systemic name will become a universal entity wherever you go, whatever the literature you cite, whichever the author you see, whichever researcher you refer, the systemic name of an enzyme molecule will remain same. It will never change. Okay, so that is the systemic name. Now, what this systemic name is and how is it given? So basically, International Union of Biochemistry and Macrobiology has constituted a commission called Enzyme Commission. As you could see here, this EC, it refers to the Enzyme Commission. Okay, what this Enzyme Commission has done, this Enzyme Commission has proposed a naming system or coding system for the enzymes. This system is referred to as Enzyme Commission number, which contains 4 different digits. Every digit denotes a particular peculiarity or characteristic of the enzyme molecule. All the 4 digits, they are separated by a dot from each other, and they will denote certain things by which you can come to know what enzyme this is. What are those digits? Let us take an example. EC, enzyme commission number M.N.O.E. Okay, so what does this denotes? First digit denotes the class of the enzyme molecule, to which class that molecule belongs. Second digit stands for the subclass of the enzyme molecule. Similarly, third digit stands for the subclass or subgroup of that enzyme molecule. And the fourth digit gives the actual identity of that enzyme molecule, that is referred to as the serial number of the particular enzyme, is depicted by this 4 digit, that is this P in the enzyme commission number. For example, we have one case here, 2.7.1.1, EC 2.7.1.1. Now this denotes the enzyme called hexokinase. Okay, hexokinase means this has got 2 components in the name. Kinase means an enzyme which brings about phosphorylation reaction. Phosphorylation reaction means a reaction in which phosphorus group is attached to the substrate molecule. Okay, that enzyme which brings about attachment or addition of phosphorus group to the substrate is always referred to as the kinase enzyme. Okay, and hexokinase means this kinase is a kinase which brings about phosphorylation of hexose sugars like glucose. Okay, now we have said that the first digit denotes the class of the enzyme molecule. So Enzyme Commission has given or has proposed 6 classes of enzymes based on the reactions they catalyze. क्लास का जो classification है, it is totally based on the kind of reaction an enzyme molecule is carrying out. First class is oxidoreductases. This class is denoted by enzyme commission number 1. Okay, so any enzyme which has got enzyme commission number EC1.X.X.X, okay, X means any number could be there. So if the first digit is 1, you will say that this enzyme is a oxidoreductase. Now, what is a oxidoreductase? Oxidoreductase is an enzyme which may either carry out oxidation reaction or a reduction reaction. Now, further, which kind of reaction the particular enzyme is carrying out, it will be explained in the subclass digit, means 1.1 or 1.2 will be there. Okay, so 1.1 will denote the oxidases, the enzyme which are bringing about oxidation reaction, and 1.2 will denote the reduction reactions, the enzymes bringing about reduction reactions. So just merely by looking at the enzyme commission number of the enzyme molecule, you can come to know what enzyme it is. Okay, by merely taking a trivial name of the enzyme, you may not know anything about the enzyme from the name. For example, say if I say amylase, you can't predict anything what this enzyme is, what this enzyme is going to do. Or if I say catalase, from the name you cannot get any information regarding the enzyme's activity. Okay, if you have some basic knowledge, previous knowledge regarding the amylase, that it brings about hydrolysis of starch, or regarding catalase, that it brings about breakage of your hydrogen peroxide, unless and until you have this prior knowledge, you cannot depict the activity, the substrate, or the product of the enzyme molecule from its trivial name. But when you refer to its systemic name or this enzyme commission number, you come to know each and everything about that. Means even if you have the number, class number of that enzyme molecule, the first information you get what kind of reaction it carries out. Enzyme commission number one means the enzyme may either be oxidizing or it may be reducing. Okay, similarly, enzyme commission number two will be including all the enzymes which are referred to as transferases. Transferases are those enzymes which bring about transfer of functional groups from one substrate molecule to another product molecule. Enzyme commission number three includes the hydrolases. All those enzymes which are bring about hydrolysis reactions are included here. Enzyme commission number four includes lyases, which bring about splitting of substrate molecule. Enzyme commission number five includes the isomerases, which brings about isomerization of the substrate molecule. Say glucose is converted to fructose. So that is the isomerization reaction, because chemically glucose and fructose both are having same chemical formula C6H12O6. But fructose is different sugar, glucose is different sugar. So that conversion where chemical properties does not change, but the conformations or the structure of the molecule changes are referred to as isomerization. Okay, and these reactions are carried out by enzymes belonging to enzyme commission number 5, isomerases. Similarly, last class, enzyme commission number 6, ligases. These are the enzymes which bring about some sort of joining reactions. Okay, such as DNA ligase is there, which brings about coupling of two DNA fragments with each other. Later on, we'll see in detail what these enzymes do. You have to study all these enzymes. You will come across these enzymes during your biochemical pathways. जब आप biochemistry pathways study करेंगे, तो बहुत सारे enzymes आपको यहाँ पर देखने के लिए मिलेंगे जो उन pathway को control करते हैं, operate करने के लिए. उसी तरह ligases या lyases, these enzymes, they are involved in genetic engineering. There also you will come to know in detail regarding these enzymes. For time being, you should just consider that enzymes, they have been classified in 6 classes: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Okay, the enzyme commission numbers will be 1, 2, 3, 4, 5, and 6. Okay, so this is the classification of the enzyme molecules. The next thing, the factors affecting the activity of the enzyme. Basically, when we refer to the activity of enzyme, so what we have said regarding the enzyme, that enzyme is a biocatalyst which accelerates the rate of reaction. Rate of reaction means the velocity at which the reaction is taking place. So when we assess the activity of a particular enzyme, so basically what we are doing, we are actually assessing the reaction velocity at which the reaction takes place. Okay, so there are many factors which affect this reaction velocity, or in other terms, you may say they affect the activity of the enzyme molecule. Those are the temperature, the pH. Okay, now this thing you have seen before also, that at certain temperatures the enzyme activity is low. When you increase the temperature up to a certain value, the activity continues increasing. When the peak is reached, and if you increase the temperature further, the activity is going to fall down. This is related to the optimum temperature required for the growth of the organism. Even you can apply this thing to humans also. You observe yourself that what happens during the winter days when temperature is say 15 or below 15 degrees centigrade, you are uncomfortable in that temperature. Okay, during the change of season, this temperature starts increasing from 15 to 20, 20 to 25. So what happens as the temperature increases, you become more comfortable in that higher temperature. But when temp up to say 30 degrees centigrade, you will be comfortable. But after that, if the temperature continues to increase, the summer days start and temperature starts building up, say it crosses 35 degrees, you can tolerate that thing. It reaches 40 degrees, you become very much uncomfortable. It becomes 45, 50, say if it becomes 60 degrees centigrade, it will be almost impossible for us to survive in that temperature. Why this thing happens? Because our enzymes, which are actually carrying out all the cellular activities in our cell, they are destined to function at a particular temperature. So when we say that we are comfortable at say 30 degrees centigrade, that means our enzymes actually are comfortable at 30 degrees centigrade. They function well at that temperature. So there will be optimum growth, highest growth of our organism at that particular temperature. Okay. After that, if you increase the temperature, what is going to happen? The enzymes will start disintegrating. It will not function properly in initial stages, and after that if further the temperature is increased, the enzyme will be disintegrated and the organism is going to die, because there are no enzymes remain inside the cell. All of you are getting my voice? Okay, so this will be the effect of temperature. Same thing is there for the effect of pH also. Now, different kind of enzymes will have different kinds of pH requirements. Similarly, the temperature requirements are also there. This image you can see we are studying two different kinds of enzymes, the pepsin and the trypsin. Now, pepsin works well under the acidic condition, whereas trypsin works well under the slightly alkaline condition. So, what you find that for pepsin, starting from the zero, the activity starts increasing up to a pH of say about 2, and after that, if pH further increases, uh, the activity starts degrading. At pH 3.5, the pepsin is totally disintegrated. There will be a damage to the enzyme molecule and activity is permanently lost. In case of trypsin, what you see, starting with 5.5, the pH increase will increase the velocity of reaction up to a pH of say 7.5, a bit alkaline pH. And if after that the pH further continues to increase, there will be a decline in the activity. And after pH 9, the enzyme will be totally disintegrated, the activity will be permanently lost, the cell will die. So different kinds of enzymes will have different optimum pH, different optimum temperatures for them. For example, say you cannot universally say that this temperature is the optimum temperature for all the enzymes, or this pH is optimum pH for all the enzymes. For example, say if you take the mesophilic enzymes, you will find that their optimum temperature is usually between 25 to 35 degrees centigrade. If you take a cyclophilic bacteria, the temperature, optimum temperature there will be between 5 to 15 degrees centigrade. If you take a thermophilic bacteria, for a thermophile, the optimum temperature will be very, very high. We have one bacteria called Thermophilus aquaticus, which is usually naturally found in the hot springs. So in Thermophilus aquaticus, the enzymes they usually are optimally active at temperatures of 90 to 100 degrees centigrade, because that bacteria is having a natural habitat in hot springs. It is usually growing at 80 or 90 degrees centigrade. So definitely its enzymes will have the same range of optimum temperature. So there will be no universal range of optimum temperature or optimum pH for a particular enzyme. It will depend on the source of the enzyme. If a enzyme belongs to a thermophile, its optimum temperature will be very high. If it belongs to a cyclophile, the temperature requirement will be similarly in the colder temperatures. Next thing, the concentration of enzymes does plays an important role, provided that if the substrate concentration is constant. Suppose in a test tube you have added 100 enzyme molecules, and if sorry substrate molecules, and if you add one enzyme molecule, what is going to happen? One enzyme will couple with one substrate, convert it to product, then will same reaction will be repeated with the second substrate. And if suppose for every substrate it requires 2 seconds, so for converting all these 100 substrate molecules, it will require 2 into 100, 200 seconds. If you increase the enzymes, instead of one, you use two enzyme molecules. Okay, the time will be reduced 100 seconds. If you add four enzyme molecules, it will be further reduced, say 25 seconds, 50 seconds, like that. And if you add 100 enzyme molecules to 100 substrate, what is going to happen? Within 2 seconds, the reaction is going to finish. But after that, say if to 100 substrate molecules, you are adding 105 enzyme molecules, no further increase. उसके बाद रिएक्शन वेलोसिटी में कोई इंक्रीज नहीं होगी. 100 एंजाइम 100 सबस्ट्रेट को 2 सेकंड में ही कन्वर्ट करेंगे, और 110 एंजाइम भी 100 सबस्ट्रेट मॉलिक्यूल को कन्वर्ट करने के लिए 2 सेकंड ही रिक्वायर्ड होंगे. दैट्स व्हाई यू फाइंड हियर दैट यहाँ पर जो आप ग्राफ देख रहे हैं, ये कॉन्स्टेंटली इस तरीके से यहाँ पर इंक्रीज होता है. इसके बाद अगर आप इसको और एंजाइम्स यहाँ पर ऐड करेंगे, तो ये कुछ इस तरीके से हो जाएगा. देयर विल बी नो फर्दर इंक्रीज इन द रेट ऑफ रिएक्शन, और वेलोसिटी ऑफ रिएक्शन में वहां पर किसी भी तरह का कोई इंक्रीज नहीं होगा. सिमिलर थिंग इज द सबस्ट्रेट कॉन्सेंट्रेशन. ना जस्ट ये कंडीशन आपको आप रिवर्स कर दे, enzymes को फिक्स रखे 100 enzyme molecules and start increasing the number of substrate molecules. Okay, now what you will observe, you will observe same kind of thing. उसी तरह की चीज आपको देखने के लिए मिलेगी, similar that आपका रेट ऑफ रिएक्शन सबस्ट्रेट के साथ साथ धीरे 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 इंक्रीज होगा. एक टाइम ऐसा आएगा जब उसके बाद आप और मजीद सबस्ट्रेट ऐड भी करें, तो उसके बाद रेट ऑफ रिएक्शन में और फर्दर कोई इंक्रीज नहीं होगी, बिकॉज एक इक्विलिब्रिया अचीव होता है. वन एंजाइम वन सबस्ट्रेट 1:1 का जब रेशियो अचीव हो जाएगा दोनों ही केसेस, तब रेट ऑफ रिएक्शन फर्दर इम्प्रूव नहीं होता है. 1:1 तक जो रेशियो था तब तक ही वहां पर इंक्रीज होगा. 1:1 का रेशियो आने तक, 1:1 से कम अगर है, वहां से 1:1 आने तक रेट ऑफ रिएक्शन वेलोसिटी आपकी इंक्रीज होगी. आफ्टर दैट if it becomes 1:2, 1:3, 1:4, 1:5, there will be no further increase. That's why you observe that after a particular period of time, when maximum rate is achieved, the reaction velocity becomes stationary. अब उसके बाद उसमें मजीद किसी भी तरह का कोई इंक्रीज यहाँ पर आपको देखने के लिए नहीं मिलता है. So this much for the day. Next portion of this particular topic will continue in your next lecture. If you have any questions, you may ask. Anybody of you have any questions? You may either open your mic and ask, or you may also write in the chat box. Both ways. Okay. And second thing, that by tomorrow you will be provided with a feedback form, a Google form हम design कर रहे हैं regarding your online lectures. So you have to give the feedback regarding the online lectures, the feasibility of time, the feasibility or convenience of this online mode of teaching. Regarding that कुछ questions होंगे 5-7 उसके अंदर. Accordingly you can place your suggestions there also. कुछ तो questions होंगे जिसका आपका objective answers देने हैं. You can also place your suggestions in that. So by tomorrow or day after tomorrow, you will be getting that thing. So as soon as you get the form, you are advised to fill up the form and submit. Okay. Meanwhile, I hope all of you have installed that application Kahoot. How many of you have installed Kahoot? Okay, Usma, one. Who is this? Paula. Okay. And who else? So I advise rest of the students who have not done this thing, who have not installed this Kahoot, kindly install it, do a bit of practice on that app, so that when we arrange online evaluation or online quizzes on microbiology or on our syllabus, it will be easier for you. Okay, so we'll stop here only. Rest of the things we'll see day after tomorrow at same time, 3:15. We'll be arranging the next lecture. Okay, thank you very much.